if you take a cell out of an individual and sort of examine that cell molecularly, the cell houses a DNA, and in fact, all the DNA necessary to encode all of the proteins that are necessary to create a cell. And that collection of DNA, as shown as chromosomes here, is collectively described as the genome. This is the entire collection of genes encoding all of the proteins necessary to create every different cell in the body. If you sort of drill down, or if you imagine expanding down into the structure of just one of these chromosomes and unpackage that, you get to the DNA itself, which is shown here. And inside the DNA, encoded by that DNA, are the individual instructions necessary to encode a particular protein. And so each one of those instructions, each one of those chapters of the book, if you will, is called a gene. And if that gene is transcribed, so this process of transcription, we can take the information encoded in the DNA and turn it via this process of transcription to mRNA and translation into the proteins that are actually encoded by this piece of DNA, this gene. So when we think about gene therapy, the concept is that by working at the DNA level, we can introduce, or as we'll describe today, subtract genetic information from the cell. And really what we're doing is essentially impacting the proteins that will really be engaged in this case, in HIV, but we're doing so at the DNA level to either add or subtract genetic information to provide the cell with some new capability that we think will assist that cell in resisting HIV infection.